welcome to educator.com. We're going to continue our discussion of large biological molecules with a lesson on nucleic acids and proteins. In the previous lecture, we talked about two other classes of large biological molecules, carbohydrates and lipids. I also introduced some basics of organic chemistry there. So if you haven't watched that lecture yet and you're not familiar with organic chemistry, you may want to watch at least the first part of that before going on to this. There are two types of nucleic acids, DNA, deoxyribonucleic acid, and ribonucleic acid. DNA contains an organism's genetic information, and that information is passed on from parent to offspring. DNA codes for proteins. In the molecular biology section, we'll talk about how DNA is synthesized, how the DNA is transcribed into RNA, and how that RNA is translated into a protein. Right now, we're just focusing on the structure of both DNA and RNA. DNA is made up of nucleotides. These are the building blocks for the DNA molecules. RNA, same thing. It's made up of monomers of nucleotides. So let's go ahead and look at the nucleotide structure. There are three parts. There's a sugar, there's a nitrogenous base, and there's a phosphate group. In both DNA and RNA, the sugars are 5-carbon sugars, so these are pentoses. However, the sugar in DNA is deoxyribose, whereas RNA contains ribose. So the name explains the difference, deoxy. Deoxyribose is missing one of the oxygens. So ribose has a hydroxyl group here, whereas DNA just has a hydrogen. So that's one difference between DNA and RNA, although their fundamental structure is the same. DNA and RNA both have nitrogenous bases attached to the sugars. And there are 5 different types of nitrogenous bases that we'll look at in the next slide. If you just consider the nitrogenous base plus the sugar in either DNA or RNA, that is called a nucleoside. With the phosphate group added, you have a nucleotide. So these nucleotides are the monomers from which the larger DNA or RNA molecule are formed. Here are shown the 5 different nitrogenous bases, and they can be classed into 2 groups. These first 2 are more complex. They have a 6-member ring fused to a 5-member ring, and these are known as purines. So these top 2 are purines. And there are 2. Adenine, it's often known by its abbreviation A, and guanine, known by just the letter G. Here, below in this row, are the pyrimidines. They're based on this 6-member ring, and these are rings that contain carbon and nitrogen. And there are 3 pyrimidines, cytosine, thymine, and uracil. One important thing to note is that thymine is found only in DNA, whereas uracil is found only in RNA. So in lieu of thymine, RNA has uracil. Otherwise, DNA and RNA have the same nitrogenous bases. A, G, and C are found in both DNA and RNA, thymine only in DNA, uracil only in RNA. One thing also to keep in mind as you're looking at structures of RNA and DNA is that in order to distinguish the carbons on the nitrogenous bases from the atoms on the sugar, the atoms on the sugars, ribose and deoxyribose, have a prime after them. So we'll sometimes be referring to like the 2' or the 3' carbon, and those refer to the different atoms on the sugar. So this prime means that we're talking about the atom on the sugar versus an atom on these nitrogenous bases. nucleotides join to form polymers that consist of a sugar phosphate backbone with the attached nitrogenous bases. So one nucleotide is linked to the next nucleotide to form what's called a polynucleotide. So the monomers are the nucleotides, and they join to form polynucleotides, and that's usually what we're talking about when we talk about DNA and RNA. The linkage that attaches these is known as a phosphodiester linkage. So remember, when you just looked at the monomers, each one of them had a sugar, a nitrogenous base, and a phosphate group. And the phosphate groups serve as the linkage between one monomer and the next monomer. We're going to talk about this synthesis in detail in the molecular biology lecture, but for right now, you should be aware that there's a directionality to DNA. And we discuss DNA and RNA as having 5' ends and 3' ends. The 5' carbon on what we call the 5' end of the polynucleotide has a phosphate group attached. So this is the 5' end. There is a phosphate group attached to the 5' carbon. This end is called the 3' end. So there's this free hydroxyl group. So here we have this phosphate group, the 5' end, and then we have the hydroxyl group attached to the 3' carbon on the 3' end. This directionality is very important because synthesis of DNA always occurs 5' to 3'. And again, we'll get into the details of that later, but right now you should just know the basics that there's a 5' end, there's a 3' end. Two adjacent nucleotides are linked via a phosphodiester linkage. And you should also be aware that this section, the sugar and the phosphate group, are known as the sugar phosphate backbone. So sugar phosphate backbone, right here, and then attached nitrogenous bases. The order of these nitrogenous bases is the genetic code. So you could have a strand of DNA that may have a nitrogenous base A attached, the next one's G, then another G, C, T, and it's these bases, it's the order of those bases that encodes the information to make a protein. And it's this information, this order of the bases, that is passed along from parent to offspring. DNA is found in a double helix structure. This double helix structure is formed by two complementary strands. These strands are also anti-parallel. So what do we mean by anti-parallel? Well, it has to do with that directionality. Let's say that this is the 5' end of one of these pieces of DNA, and we follow this down, and it's going to end here at the 3' end. The other strand is going to have the opposite orientation, where it starts with the 3' end up here and then ends at the 5' end down here. So we say that these two strands are anti-parallel. So they are complementary and they are anti-parallel. So the 5' to 3' versus 3' to 5' explains anti-parallel. What does complementary mean? Well, that has to do with what's called base pairing. Remember that in DNA, there's no uracil used. We have A, G, C, and T. Adenine pairs with thymine, and this would be called a base pair. And G, guanine, pairs with cytosine. Here's a sugar phosphate backbone shown with the purple. And then the nitrogenous bases, A, G, C, and T, actually stick inward towards the center, or towards the middle of this double helix. This double helix structure was actually famous discovery by Watson and Crick in the 1950s. And so here we have the sugar phosphate backbone. And then let's say this nitrogenous base here is A. And T, C, G, A, T. The complementary strand, since A pairs with T, this complementary strand is going to have a T. T pairs with A, the complementary strand will have an A. C is going to pair up with G. So you're going to have a G here. G, C, A pairs up with T, T pairs up with A, and so on. So complementary refers to the fact that the base pair on the strand, on the second strand, matches with the base on the first strand. Now, hydrogen bonding occurs between these base pairs, and that's what holds this helix in its structure. And you'll notice up here that I showed 3 dotted lines between G and C, whereas there's only 2 dotted lines between A and T. And that's because AT forms 2 hydrogen bonds with each other, whereas G and C form 3 hydrogen bonds with each other. Okay, so important points to remember is that DNA is a 5' end and a 3' end. Nucleotides are linked by a phosphodiester linkage. Their structure is such that there is a DNA double helix that's formed between 2 complementary anti-parallel strands. Each strand is paired so that it has a matching base pair. A always pairs with T, G always pairs with C, and these nitrogenous bases, these base pairs, form hydrogen bonds that keep this double helix structure in place. RNA is single-stranded. In biology, form follows function, and you'll see how this double helix is very important in the synthesis of DNA and in producing DNA that has as few mistakes as possible so that genetic mistakes or mutations are not introduced. So we'll revisit this double helix structure later on with a focus on DNA synthesis, transcription, and translation. The next large biomolecule that we'll talk about is proteins. Amino acids are the building blocks of proteins, and these join to form polypeptides. So remember that DNA contain genes, and these genes encode proteins. Proteins are extremely important to living organisms because they make up structural elements such as our muscles. They facilitate reactions in the form of enzymes, which we'll discuss later. They're involved in cell signaling, they're involved in cell growth, and the repair of cells. So proteins are fundamental to life. There are 20 main or common amino acids. You don't have to know each of these. You just need to know the general structure of amino acids and some different classifications of the amino acids. So let's look at just a generalized amino acid. You're going to see that it has certain groups. Here, it has an amino group. On the other side, it has a carboxyl group. In the center is a carbon atom, so bonded to the carboxyl group, the amino group, a hydrogen, and then what we call an R group. The R groups are different, so each of the 20 amino acids has a different R group. It could be as simple as just hydrogen. That would be just glycine, structurally the simplest amino acid, or it could have more complex functional group like shown here with asparagine, or even more complex than that. You might see amino acids also written, instead of NH2, you might see this shown as NH3 plus and the carboxyl group as COO minus. And that's because in water, the amino group picks up a hydrogen ion and therefore acts as a base. And the carboxyl group loses a hydrogen ion and therefore acts as an acid. You're also going to notice that there are 3-letter abbreviations for the amino acids. So you might see instead of asparagine written out, ASN. Or for example, leucine is another amino acid that's abbreviated as LEU, and glutamine, GLN. There are also 1-letter abbreviations for the amino acids. Let's talk a little bit more about these side chains. The side chains can be grouped such that they are nonpolar, they are polar, they are acidic, or they are basic. First, let's just talk about nonpolar. Glycine has an R group that is just a hydrogen atom. That's going to be an example of a nonpolar amino acid. A polar amino acid might have, for example, hydroxyl groups on it, or some other polar functional groups. Asparagine is actually considered a polar amino acid. So this one's polar. Acidic and basic are a little bit more complicated. When we talk about acidic or basic amino acids, we're not talking about the amino group here or the carboxyl group here. We're talking about the R group or the side chain. If you think about it, in solution, this is going to pick up a hydrogen ion, this is going to lose one. They're going to essentially neutralize each other. So the amino acids overall, in the different forms that they're in solution, the net is going to be that they're neutral. However, if there is a carboxyl group on the side chain, if the side chain has a carboxyl group, then you would have an acidic amino acid. If the side chain contains an amino group, then you would have a basic amino acid. So again, we're not looking at this. When we talk about polar or nonpolar amino acids, we're talking just about what's going on with the side chain. I want to point this out. This is a bit of an exception. Glutamine and asparagine are both polar. They are not basic. Even though you see this NH2 here and you might think, oh, an amino group. This particular construction where you have C double bonded to O linked to NH2 is something called an amide group. And this is a bit different. It's not actually basic. If you had this NH2 but it wasn't linked to the C double bond O, then I would say, okay, you have a basic amino acid. But this particular group together actually is not a basic side chain. Glutamine has this group. Asparagine has this group. These two are classed as polar, not as basic. Aspartic acid is a good example of an acidic amino acid. The side chain contains COOH, this carboxyl group, and at cellular pH, this becomes ionized to COO minus, and therefore this is an acidic amino acid. It tells you so in its name. Okay, so we talked about amino acids. These are the building blocks for proteins. Amino acids can be joined together through the formation of what's called a peptide bond. A peptide bond is yet another dehydration reaction. We discussed that in previous lecture, where a dehydration reaction is a reaction where water is removed. So we lose a water. So recall that the basic structure of an amino acid would be NH2, central carbon, an R group, hydrogen, and then we have this carboxyl group, COOH. An adjacent amino acid would have the same structure. So now we're just going to do COOH over here, R here. Here we have the NH2, and here we have H. Actually, let's write this as NHH. This peptide formation, again, is through a dehydration reaction. So water's going to be lost. So if an OH is removed from here, and an H is removed from here, you're going to end up with H2O removed, and that'll leave this CO, which is this, becomes double bond, and this nitrogen, which is here, bonded together. So this carbon from the carboxyl group bonds to the nitrogen from the amino group, and water is lost. Again, COOH and NH2, loss of an OH group from the carboxyl, a hydrogen from the amino group, leaving behind C double bonded to O, and then the C bonds to the nitrogen on the adjacent amino acid, forming a peptide bond. What's shown here is a dipeptide. It's 2 amino acids. 3 or more amino acids are a polypeptide chain. And a polypeptide chain can be very short, just a few amino acids. It can be 1,000 amino acids long. The primary structure of a protein is the amino acid sequence of its polypeptide chain. There are multiple levels of structure in a protein. There are 3 to 4 depending on the type of protein: primary, secondary, tertiary, and quaternary. The primary structure is just the order of the amino acids. If it's leucine and glycine, glutamic acid, then an asparagine, another asparagine, that would be giving the primary structure of the protein. Now, polypeptides are not the same as proteins. Polypeptides are just chains of amino acids. They're in a particular order, but if they're unfolded, they're just chains, they're polypeptides. In order to actually be a protein, the polypeptide chain or chains, sometimes there's more than one chain in a protein, need to be folded in a unique 3-dimensional conformation, then that's a protein. So once you just have a bunch of amino acids linked, it's not a protein yet. It's a protein once it's folded. The primary structure is actually what determines this folding, although nobody's quite sure how. And the primary structure of a protein was first elucidated by Fred Sanger at Cambridge, and he and his lab actually sequenced insulin. Remember that insulin is a hormone secreted by the pancreas, and it's vital in controlling glucose levels in the bloodstream. Without enough insulin, a person becomes diabetic. Another medical example of the importance of the primary structure of a protein is the disease sickle cell anemia. This disease is caused by the substitution of a single amino acid, valine. Instead of valine, valine is substituted for glutamic acid. So it's a valine substitution. That change of that one amino acid to a different amino acid causes problems in the structure of hemoglobin. So hemoglobin is found in red blood cells that carries oxygen. The structure of the hemoglobin is abnormal in people with sickle cell anemia. And the result is that it actually causes the red blood cells to form a sickle shape. And that sickle shape, those sickle-shaped cells tend to clump up. So red blood cells are normally a biconcave disc. They move nicely through even small vessels like capillaries. With sickle cell anemia, these cells form a sickle shape, they clump up. That causes poor circulation to various parts of the body and symptoms. So the primary structure of a protein is very important in determining the secondary and tertiary structure and the function of the protein. So we went through this first level of structure. Let's go into more detail about the other levels. The secondary structure of a protein is a result of hydrogen bonding between different regions of the polypeptide backbone. When I say polypeptide backbone, that means not the side chain. It means the other parts. The 2 main protein secondary structures are alpha helices and beta pleated sheets. So let's go back to how these are formed. There are certain regions on a polypeptide strand that are repeated, and particular repeated regions allow for hydrogen bondings. This bonding occurs between the electronegative oxygen atoms and the hydrogen atoms attached to the nitrogen. So remember your basic amino acid structure. There's an R group, there's a carboxyl group, there's hydrogen, and there's an amino group. And the hydrogen on the amino group is attracted to the electronegative oxygen atoms and these carboxyl groups. So this would cause hydrogen bonding between one amino acid and another nearby amino acid. So we're not talking about the side chains. We're just talking about the backbone structure, which is this part. So the electronegative oxygen and hydrogen atoms attached to nitrogen can form hydrogen bonds. So if there are certain repeated regions, particular types of repeated regions, you can end up with either this helix shape, it's like a spiral, or what's called a beta pleated sheet. And it's like if you took a piece of paper or something and folded it up. So this is the alpha helix and beta pleated sheet. So the important thing to remember about secondary structure is that the hydrogen bonding is between the polypeptide backbones, not the side chains, and that the two main types of secondary structure are alpha helices and beta pleated sheets. Again, one hydrogen bond is not that strong, but many hydrogen bonds together could hold a structure in place and stabilize it. The next level of structure is tertiary structure, and tertiary structure is the overall three-dimensional shape of the protein. So this shows a protein that might be described as a globular protein. It's shaped like a globe. An example would be hemoglobin. Within this overall three-dimensional or tertiary structure, certain regions may have an alpha helix. Maybe this region over here is beta pleated sheet, and this is beta pleated sheet, and this is neither. And then there's another alpha helix here and here. So the entire polypeptide doesn't usually form one big alpha helix or beta pleated sheet. Often there's just little regions of each in certain areas of the protein. And then those regions, along with the rest of the protein, fold into a unique shape. And the shape is extremely important to the protein's function. When we talk about enzymes, you'll see that the shape of the enzyme is crucial into it being able to facilitate a reaction. There are 5 types of interactions that form and maintain the tertiary structure. The first one is hydrogen bonding, but this time it's of those side groups. Remember that hydrogen bonding between the main polypeptide backbone will form secondary structures such as alpha helices and beta pleated sheets. Those amino acids that contain hydroxyl groups or amino groups, for example, can hydrogen bond and help to stabilize the three-dimensional structure. The second type of bond is ionic bonds. Remember that there are certain charged side groups. We talked about the fact that there are some amino acids that are acidic and some that are basic. So those are charged, and they can form ionic bonds with each other. The third type is known as a disulfide bridge. Cysteine is an amino acid that contains SH on its side chain. 2 cysteine molecules can each lose a hydrogen and form what's called a disulfide bridge or bond. So if I have 2 cysteines near each other, they each lose a hydrogen and they form this disulfide bridge or disulfide bond. And this is a covalent bond, but it's still crucial in forming the tertiary structure. The fourth type is hydrophobic interactions. And what this refers to is that nonpolar or amino acids with nonpolar side chains often end up clustered in a central part or in the middle of the protein structure. And this is because those amino acids that have polar side chains can form hydrogen bonds with water. So when protein's in solution, the side chains that are polar interact with the water molecules, they form hydrogen, and then the nonpolar ones pretty much just end up excluded from that and pushed to the center. And when these nonpolar ones are near each other, they can form various bonds and nonpolar interactions. There's one type of interaction between nonpolar molecules that are called van der Waal reactions. These are another type of weak bond, and these form between nonpolar molecules. So again, the tertiary structure is the overall shape of the protein. It involves bending and folding, and there are 5 types of interactions that form and maintain the tertiary structure: hydrogen bonding between the side groups, ionic bonds between side groups that are charged, disulfide bridges between cysteine molecules, hydrophobic interactions, and finally, van der Waals interactions. There's one final level of structure only for certain proteins, and this is quaternary structure. There are certain proteins that are formed from more than one polypeptide chain, and the interaction between multiple chains in a multi-subunit protein is known as quaternary structure. An excellent example is hemoglobin. Hemoglobin has 4 chains, 4 polypeptide chains that form the 4 subunits of hemoglobin. And not only does each of these chains have a certain tertiary structure, but all 4 of the chains come together in an overall structure known as a quaternary structure. When you think about protein folding, you need to be aware that certain conditions affect folding. There's an optimal temperature for the folding of proteins. There's an optimal pH and osmolarity or salt concentration. If, for example, temperatures change, if a protein's heated up, it melts or unfolds, and we call this denatures. When a protein denatures, that means that it unfolds. If the pH is changed, if you take a solution and add a bunch of salt to it, the proteins in it could also denature. So proteins function optimally at certain conditions depending on that protein. Like in our bodies, the pH, most areas of our body, is about 7.4. So most of the proteins in our body function best at that pH and at our body temperature. If a protein denatures, since form follows function, it's not going to function very well. Now let's go ahead and review some examples focusing on nucleic acids and proteins. Write the complementary strand for a strand of DNA with the following sequence. All right, this shows the 5' end and the 3' end. And then it shows a sequence GCCAGTTCAAG for DNA. So this is the different nitrogenous bases. The first thing in a question like this is to remember that the 2 strands are anti-parallel. So right away, as part of the answer, you should always write in the 3' and the 5', and they're going to be opposite because it's anti-parallel. So the strand is going to be anti-parallel and complementary. Remember that G forms 3 hydrogen bonds with C, and A forms 2 hydrogen bonds with T. So these are the ones that pair up. Therefore, when there's a G, it's going to pair up with a C. A C will pair up with a G, again with a G. Here I have an A, it pairs with T. G pairs with C. 2 T's, these each pair with A. C pairs with G. A's pair with T. And finally, G and C. So the complementary strand is going to be 3' to 5'. The G's will be paired with C's. C's will be paired with G's. A and T go together, and T and A. So this is the complementary strand for this strand of DNA. Example 2, lists 2 differences between DNA and RNA. Well, recall that RNA contains uracil, DNA contains thymine. That's one difference. Second difference, RNA is single-stranded. DNA is double-stranded. And they only ask for 2, but the third difference is that RNA, the sugar for RNA is ribose, whereas the sugar for DNA is deoxyribose. So here's 3 differences. Any 2 would have sufficed to answer the question. Difference in nitrogenous bases, single versus double-stranded, and ribose sugar versus deoxyribose sugar. Is each of the following amino acids polar, nonpolar, acidic, or basic? To determine that, remember you're just going to look at the side chains. The side chains right here, here, and this whole region here. I am not going to pay attention to this carboxyl or amino group that's present on the backbone. Right here, this is the simplest amino acid I mentioned before, which is glycine. And glycine is non-polar. It just has a hydrogen. This second amino acid is serine. Serine, you see, has a hydroxyl group, which is polar. So this is a polar amino acid. Lysine is a little bit more complicated, and you'll notice that it has an amino group. When this is put in solution, this amino group is going to pick up a hydrogen ion and become NH3+. Therefore, it is basic. So we have 3 amino acids, 3 different categories: nonpolar, hydrogen side chain. Serine is polar with the hydroxyl group. And finally, lysine, which is a basic amino acid. List 4 types of interactions that maintain the tertiary structure of a protein. The first one is hydrogen bonding. And remember that this is hydrogen bonding between the side groups or the R groups, not between the backbone. Second type is hydrophobic interactions. These occur as a result of amino acids with hydrophobic side chains being pushed towards the center of the protein, being excluded from the hydrogen bonding of water and the polar side chains of some of the other amino acids. So these are hydrophobic interactions. A type of weak interaction between nonpolar side chains is called van der Waals interactions. If there are some cysteine-containing amino acids that are cysteine, they contain sulfur, then there may also be disulfide bridges that help to maintain this tertiary structure. So this was 4, but a 5th one is ionic bonds between amino acids that are charged. So here's 5. Any 4 of these would answer the question of the 4 types of interactions that maintain the tertiary structure of a protein. That concludes this lecture on educator.com. Thanks for visiting.